My dear viewers, welcome to Biology at Priyajit. In today's class, we dive into the fascinating world of eyes in insects, one of the most advanced and diverse visual systems in the animal kingdom. You will learn how insects use compound eyes, ocelli, stemmata, and even dermal receptors to see, survive, hunt, escape predators, and communicate. We will explore how thousands of ommatidia come together to form clear mosaic vision, how nocturnal insects create superposition image, and why insect vision is a key evolutionary success story. By the end of this session, you will understand how insect eyes are built, how they function, and how their incredible adaptations support life in every environment. Importance of eyes or vision in evolution. Vision played a major role in the evolution of animals, helps in searching for mates and recognizing environmental conditions. Predator-prey interactions increased the need to see and avoid being seen. Camouflage, protective cells, and survival strategies evolved due to vision pressure. According to the light switch theory, advanced vision improved survival and shaped evolution. Insect vision basics. Many insects or most of the insects have highly developed visual systems. Only a few subterranean and parasitic insects are blind. So the vision requires first of all a lens to focus light, photoreceptors to capture light using pigments, and nerves and brain to process visual signals. Rhabdom is the light receiving structure made up of retinula cells with visual pigments. Light changes pigment structure. It will convert into an electrical signal which reaches brain via the synapse. Two types of insect eyes. First one is a compound eye, which is the main visual organs in adults, and simple eyes, that is a single chambered and found mostly in larvae and immatures. Comparison criteria for insect eyes. Visual systems vary mainly in resolving power and light sensitivity. Resolving power, clarity and detail of the image, and light sensitivity provide ability to see in dim light. Other light detecting systems, insect detect light through dermal detection, stemmata, ocelli and compound eyes. Dermal detection. Some insects can detect light through body surface. Example, yellow mealworm or tenebrae molitor larva. So though no lens or focusing structures are present in yellow mealworm, epidermal cells are very much light sensitive. Light response continues even after covering eyes, which can be seen in cockroach and caterpillars. Cave insects, which are blind in nature, without eyes they can respond to light. So aphids have light sensitive cells in their brain and detect day length, and they control their process of reproduction. And butterflies have photoreceptors on genitalia. They help during copulation. Examples: papilio, which have two receptors per body side, each with a single neuron. So yellow mealworm is tenebrae molitor larva. They uh, have a dermal detection property to visualize their surroundings. Stemmata or larval ocelli, they found in holometabolous larvae. We know what is holometabolous insect, in life cycle of which have four different stages like egg, larva, pupa and adult. So example is the caterpillar. Caterpillar is the larval stages of the butterfly. So butterfly is a holometabolous insect. So there stemmata or larval ocelli located on the larval head, and each structure has many photoreceptors and nerve cells. It has a cuticular lens over crystalline body. The lens focuses light on rhabdom. They have advanced larvae, sawfly, tiger beetle. They show complex stemmata. So two-layered lens form an image on retina with many rhabdoms. They are designed for high light sensitivity and low resolving power. This is a simple stemmata of a lepidopteran larva. Ocelli, they are found in adults and some nymphs. They have three on head top. Embryologically, they are similar to stemmata. A transparent cuticle acts as lens. So light passes to retina with many rhabdoms. Retinular cell groups are surrounded by pigment cells. The focal plane below retina and influences blurred image formation. So axons connect to brain neurons and their important functions are detect light intensity, detect horizon and aid flight stability, and which are very sensitive to low light. So that is not used for detailed high resolution image. This is the light adapted median ocelli of a locust. Compound eyes first appeared over 500 million years ago, seen in trilobites, the most advanced visual organs in insects. They are found in adults and nymphs. They provide nearly 360 vision, made up of optical units such as ommatidia. Which have more ommatidia, they have greater resolution in their eye. So example, dragonfly, which have 30,000 ommatidia per eye, and each ommatidia forms one pixel combined image or mosaic. The structure of ommatidia: cornea, which is a transparent outer lens, refracts the light rays. Corneagen cell, there are two cells secrete cornea and latter form primary pigment cells. Vitreolar cone cells, four transparent cells around crystalline cone. The crystalline cone is a transparent structure which focuses light surrounded by primary pigment sheath. Retinular cell, there are three elongated photoreceptor cells. They contain microvilli forming rhabdomer, and rhabdomer fusion causes rhabdom, which is a central photosensitive rod. Sensory fibers connect to protocerebrum. Pigment cells, seven secondary pigment cells isolate each ommatidium laterally. Two types of images formed by insect compound eyes. First one is the apposition image, which occurs during diurnal insect. This is the image. First one is the image formation, apposition image. It is found in insects active in bright light or housefly. The retinulae lie near crystalline cone and which is surrounded by pigment, prevents light mixing. Each ommatidium receives light only from its own lens. So individual tiny images are formed and side by side mosaic is formation. Result, it formed clear, sharp image and mosaic vision. Superposition image or nocturnal insects, they are found in night active insects such as cockroach. The retinula placed deep below cone and the pigment sheath absent, which is responsible for spreading light. Rhabdom receives light from multiple ommatidia, which causes overlapping, bright and blurred image. This is the formation of superposition image. So this is the table showing the differences of the image formation, that is apposition image and superposition image, which varies on their depends on their habitat. There are differences in pigment isolation, light intake, image quality and sensitivity. So to conclude, insects possess some of the most remarkable visual systems in the animal world. Their compound eyes, ocelli, stemmata and even dermal light receptors work together to help them navigate, locate, escape predators, communicate and survive across diverse environments. From clear mosaic vision in diurnal insects to superposition images in nocturnal form, these eyes reflect millions of years of evolutionary adaptation and success. Understanding insect eyes not only reveals how they see the world but also shows how structure and function support survival. Hope today's class made the topic clear and exciting. Stay curious and keep exploring biology. Thank you.